So this is, in the spirit of the last speaker, this entire talk is largely based around unpublished data that I generated in the plane over here. It's a long ride. So I just want to start off at the end. I'd like to thank and point out some of the people that have been pivotal in what I'm about to tell you. Mandy Ballinger, who is the driver not only behind the International Sarcoma Kindred study, but also our studies in somatic mutation testing and molecular therapeutics, our own version of the MATCH program in Australia. This fellow here, Mark Benesi, has done a lot of the bioinformatics, and so has Emma Rath. But we have a team that's quite committed to genomic cancer medicine, and you may recognize two alumni from Toronto. So the ISCS has been around now for almost a decade, and it started off in Australia, but is now operating at 23 sites in 7 countries. I'm very proud of this because all of those sites are contributing without financial support to a concept which in 2008 seemed like a good idea, but which in 2018, with the advent of whole genome sequencing at population scale, has really been quite fundamental. And the fundamental principle of the study is to take sarcoma patients, no matter what their family history or other circumstances, and enroll them consecutively onto a program which attempts to understand, using the best tools available, what the genetic etiology behind their condition is. And in effect, it combines both the principles of GWAS, that is a study of the population of unrelated individuals, with the strengths of linkage, that is, we have pedigree data on each of these cases. It's an extremely diverse population, and that's why it's now 2,500 cases, and we intend to keep it rising until we have enough within each of these categories to be able to understand histotype-specific genetic etiology. But you can see already that sarcomas particularly affect a younger population, particularly the bone cancers, and we have a number of people in the cohort who have benign connective tissue tumours as well. In total, we're up to 2,645 probands. We have bloods on 2,366 relatives. We have a number of controls, and if I've got a message to send, it's that controls are absolutely pivotal to understanding what genetic variation means. We're beginning to tackle the sequencing of the tumour blocks to understand what the tumours can tell us about the underlying things that we discover in the germline, but we've also spent a lot of time collecting information from the subjects about what genetics means to them, anticipating a time when their opinions about this matter may well be important to returning the results to them. And in total, the thing I find staggering about this study is that we have information on almost 100,000 individuals, all pivoting on the hub of the fact that they've got this rare set of diseases called sarcomas. It's been a very productive study. There's been an enormous range of papers that have come out over the past decade as the data matures, but the pivotal paper that we published was in 2016 on the first 1,162 subjects with a series of controls. And the things that I took away from that study were that 26% of the sarcoma population that walk in through the doors into our clinics, and I'm a sarcoma oncologist, that's where I come from. I'm not a professional geneticist or a genomicist or anything of that kind, but 1/4 of these people have really quite remarkable family histories, something that I now incorporate into my clinical practice. And fully 15% have multiple primary cancers, which is consistent with, again, strong genetic or environmental exposure. And at a population with an age of 46 years, that is already 50% higher than the general population at large in Australia, where 10% multiple primary cancers over a lifespan is consistent with a median age of onset of 67 years. So this is a population with a very high incidence of both multiple primary cancers and suggestive of genetic etiology. Now, we took a very simple panel, and the first panel that we did was 72 genes. These genes are well studied in the context of breast and bowel and all the other cancers that have been well studied in clinical genetics, and just ask what we'd find. And You'll see here that p53 stands out. Across all these heterogeneous tumours, there's actually a 12-fold odds ratio of carrying a p53 mutation, and then a whole lot of other genes out of the 72 which reach statistical significance, including genes involved in DNA damage repair upstream of p53, as well as that great mimic BRCA2, and interestingly, a gene which I won't go into any further called ERCC2. Now, this is where we started with doing this study, but we've just completed the sequencing of 1,100 cases out of the 2,500 with whole genome sequencing, along with some controls, and that's what I'll talk to you about today. We're fortunate having the Southern Hemisphere's largest sequencing facility, which makes all this technically possible. So the cohort that we've just sequenced is now 1,111 individuals with a median age at cancer onset of 50 years, age at first sarcoma, typically a bit later because sarcomas often arise as second malignancy in this population. The majority of these cases were actually selected to be Caucasian, non-Finnish European in origin, and the reason we did this was because of the importance of having matched controls, and I'll come back to that in a minute. The number of cancers per individual, you see 18% of this cohort had multiple primary cancers, and you see a very broad range of tumours as outlined here. Just draw you to this tiny little figure, 26 different types of sarcoma outside of the top 12 or so that I've listed there. So what about Li-Fraumeni syndrome? 138 individuals, or about 12.5%, had a family history consistent with Chompré or classic Li-Fraumeni, with a median age of first cancer onset of 35 years, although very broad range. Of that group, 33% had 2 or more cancers, which is a 1.5 to 2.5-fold increase over the cohort as a whole. And if you looked at the group who had a p53 mutation, we found 12 individuals with p53 mutation that was either nonsense or previously described as pathogenic in ClinVar. Then the age of onset dropped slightly, but the multiple primary numbers increased 12-fold in total, as you might expect. And this is what that looks like. If you look at the Chompré LFS group and the group that had p53 mutations, of the 17 who had p53 mutations in total, 5 did not have a family history consistent with LFS, but 12 did. And that leaves an enormous group, of course, that's unexplained. I'll come back to that in a minute. Of the Chompré group, there were 124 individuals, 42 with multiple primaries, and 7 of those had p53 mutations. We had a higher rate of p53 mutations, gratifyingly, amongst the 14 with classic LFS, suggesting, as we've known for some time, that classic criteria are better predictors of the carriage of p53 mutations at a time when perhaps that sort of clinical decision-making is less important. To go into that in a bit more detail, these are the types of mutations we're describing. Some of them are fairly soft, but you see the criteria that we've used to pick them out of the cohort. And these are 5 individuals who did not have a classic LFS. And you see that R158H and V157I are pretty soft and maybe atypical, but there are some here with what look like pretty convincing and mutations frequently found in cosmic. Of the 126 cases with Chompré LFS, 91% were not explained by looking at p53 alone. And so I looked into the data on the Sanger core gene, cancer gene census, which is about 380 genes, a bit of an expansion on the 72. And you see here that there is a long tail of very atypical patterns. So some of these people have digenic effects, and I can't emphasize how important I think this is going to be, not just in respect of p53 hypomorphic alleles with XAF1, but combinations of 2 different genes with good-going C45 mutations that are occurring within the one individual, including this individual here who's got an MEN1 and an MSH6 digenic mutations, ATM homozygotes, and so forth, as well as a long tail of individuals with BRCA2, P10, and the like mutations. Now, these are the criteria which we use to include these variants. The median age of cancer onset in this group is still young, and you'll notice the difference between the recessive or polygenic effects, and that the first-degree relatives in these families have a much lower chance of having cancer than those with the dominant genes, which is what you might expect. If you have a polygenic or a digenic or a recessive effect, you'd expect the risk to first-degree relatives to be typically lower, and that is a pattern that we have observed previously. So that's just a description, very brief description of what we've found. What I want to talk about now is the case control design. It's a very interesting area statistically. So what we've done in this analysis I'm about to discuss with you is to compare it to a group of 2,500 well Australian elderly individuals who have no cancer, also no cardiovascular disease, no dementia. We've designed them as a universal reference cohort. And the argument here is that by taking a group who are elderly without having a cancer phenotype, you're going to be depleting that population specifically of pathogenic alleles. Or in other words, it would be unlikely that any allele found in this group is going to be a cause of Li-Fraumeni syndrome in a young population. This is called, or an extension of, application of the concept of extreme phenotype sampling. So a little bit about the control group, which is itself very interesting from a genetic point of view. Here we have 2,500 individuals, all of whom are over 70 years of age at the time of data collection and up as high as 95 years of age. You can see the majority of them are non-Finnish European and therefore designed to match the ISCS cases. And this is industrial scale genomics. So the figure that I love about this most of all is that Mark Penese in November of last year had already spent more than 3.2 million CPU hours analysing this data. And at 1 gigabit per second, a full download of just the control set alone would take 4 months at 100% efficiency. I just can't get over it. These are landscapes of extraordinary richness and complexity. And I won't have a chance to talk to you about this, but when you look into the blood genomes, you don't just get a measure of germline inherited variation, you get clonal hematopoiesis, the sort of things that we heard Adam speaking about a few minutes ago. And that in itself gives you, in effect, an endophenotype in the peripheral blood that you can correlate with germline susceptibility. You can see measures as people age, particularly in the elderly, of essentially somatic mutations, including presence of good-going p53 mutations which were not inherited. Complexity I won't go into any further. This just shows some of the principles of extreme phenotype sampling, just looking at polygenic risk score by comparison with a group of 270 prostate cancer cases that we happen to have lying around. And this is the nomad set of controls showing the polygenic risk score distribution, showing an overlap with our cancer cases. And this shows the position of the deplete MGRB cohort. And this graph simply indicates the increase in power of using an extreme phenotype elderly control compared to a cross-sectional control such as is frequently used. Nomads frequently used for. You can use some of this to look at this case control design to look at non-coding variation where you don't have the advantage of predictive algorithms or ClinVar to help you. And I'll just show you a brief example of how one might use it by looking at p53 itself. Since it was the most frequently mutated gene in this cohort, we reasoned that there might be non-coding variations intronically within the locus that might be enriched in the case series but not in the controls. And so here you see the exons and the mutations that are pathogenic in the exons and some splice variants also in there that are just adjacent to the exons by comparison with an MGRB group that was at that point twice the size. You can see there's many more of the coding mutations than you see in the MGRB. But what I'd like to point out is that there's a statistically enriched ratio of rare singleton SNPs in the non-coding regions that cluster around the coding exons of p53, and that at least gives us a hint that that variation might itself indirectly also contribute in toto to the p53 associated phenotypes in the ISCS cohort. I'll leave it at that for now. But I wanted to talk to you about the burden of rare pathogenic variants in the cohort now using the case control designs. And again, in this data analysis I'm showing you, these are just the most pathogenic extreme end of the mutations. As you'd expect, out of the 380 genes in ranked order, p53 mutations stood out with an odds ratio of 16-fold, NF1, unsurprisingly, at 13-fold. But then the next gene I didn't expect to see, and that's POT1 with an odds ratio of 13-fold. It's got daylight between it and ERCC3 down below, where the numbers in this case control design are too small really to go any further. So POT1 was not a gene which I'd thought of as being associated with sarcoma syndrome. So what is it? It's a gene called protection of telomeres 1. It's a part of the sheltrin complex that caps the frayed ends of the shoelaces of chromosomes to stop them from fraying. And it's that fraying which can occur with conditions like dyskeratosis congenita, other syndromes associated with cancer susceptibility in children. And there's numbers of components like DKC1, which is dyskeratosis congenita 1, but a number of genes in this complex. And when you get disruption of this complex, you get increased chromosomal instability, which is a feature of about 80% of sarcomas, and indeed a feature of Li-Fraumeni syndrome. Disruption of this complex activates an ATR and CHEK1 dependent signal to p53, just to tie these things together. The fraying of telomeric ends is probably pretty important conceptually in a number of sarcomas, but particularly well and dedifferentiated liposarcoma, which for the aficionados is a disease of breakage-fusion bridge, which in turn is a problem of naked DNA ends at the telomeres. In fact, there has been a paper published in Nature Communications describing a mutation in POT1 associated with angiosarcoma and p53 families. The gene itself operates in an autosomal dominant fashion. It's associated with melanoma-prone syndromes, gliomas, CLL, and this one case of angiosarcoma. So in the ISCs, the interesting thing about this POT1-associated group, of which we had 14 in total using our most lax criteria, was that it wasn't associated with early onset disease at all. In fact, it was associated with disease onset at 62 years, and that for me is something I find puzzling. There are no patterns, but 10 out of 32 first-degree relatives were affected with cancer, suggesting an autosomal dominant penetrance, and of these families, 6 had excessive melanoma. 2 individuals, probands, had multiple melanomas and sarcoma, and 3 individuals had angiosarcomas. There were a further 7 individuals who had chromosomally unstable, undifferentiated pleomorphic sarcomas or leiomyosarcomas, absolutely consistent with a phenotype you might expect of fraying telomeric ends. And this just shows you what two of the pedigrees looked like. So this is the proband, thyroid cancer at 58, sarcoma at 62, and then this range of melanomas. I remember this fellow, he was one of my patients, and we were wondering about what his explanation was. I looked at my 72 gene panel and didn't find an explanation, and it turns out that he has a canonical mutation at glycine 94 to glutamine. And you can see all the melanomas in other individuals in this family. And likewise, this individual had a sarcoma, again, a chromosomally unstable tumour at the age of 50, who has a brother who has not 1, but in fact 6 melanomas over the course of a lifespan. So this all fits with what we understand about the POT1 syndrome. But what I hadn't appreciated is that its richness in the cohort should be up in the group along with NF1 and P53. And I think this is a very striking finding. It's our plan at this point to go and look at the entire sheltrin complex, because one of the virtues of a whole genome is that one can take an unbiased, one could say almost a proteomic approach to the associated interactors of genes which nucleate these pathways. And the second, so I'll just summarize what I've told you today. POT1 is important and common in sarcomas. It doesn't explain early onset sarcomas like Li-Fraumeni syndrome. There's only one family with LFS type pattern in it. Angiosarcomas did come up, and it's linked to melanoma, and there are other components of this complex that we should look at. There is, however, a long tail of p53 mimics, including many polygenic cases, and I think there is almost certain in a complex analysis of this cohort to be common and rare variants that contribute towards those cases which we don't yet fully understand, particularly with Chomprey criteria, where the vertical transmission in family members is less likely to be a criterion suggestive of a dominant effect and maybe more consistent with the polygenic effect. And I think the age of the panel test may well be designed to solve these problems. And that should leave me with 2 minutes to answer the many questions I need to have asked of me. Thank you. And I welcome anybody who wanted to use the cohort. It's intended as a public asset. Thierry?
Hi, very nice talk. As highlighted by several people in Science and Nature, based on this type of studies, because you are looking for enrichment of rare variation between case and control. Since you are performing an exome analysis, you should use a Bonferroni correction because you are performing 20,000 independent tests. In other words, you should reach a p-value on 10 minus 6 before the conclusion of a significant assay. And correct me if I have not correctly seen the results, but you don't reach this significancy.
No, absolutely you're correct. There are several things I'd say. The first is that we don't rely upon a classic GWAS unbiased approach. We know a lot about these genes, and the fact that p53 comes out in the top of the list is surprising given what we know about the Li-Fraumeni syndrome. In fact, I use and calibrate the sort of variants that I would select on the basis of P53 to select unbiased the remainder of the list. So we use a lot of clinical information to make these judgments. That's why I reported the family histories and they're consistent with POT1. The second thing is that you can take approach of aggregating those variants. So you could aggregate them across the pathway and then do the same sort of analysis, which gives you more statistical power to do the comparisons. And then finally, I think replication sets. When we did the Lancet paper, we went to great lengths to replicate all our findings in an independent test set. And I think that, again, is going to be very important here. We're going to go to Sharon and other holders of large data sets to verify the sort of enrichment we're seeing in an independent test set before we'd expect anybody to believe these results. It's a good question.
One more question, Sharon. Yeah.
Not the Sharon he was just referring to, but
Plon, not Savage.
right.
Although
so
you can.
I was going to say that POT1 was also previously reported as a familial melanoma gene, and I
Yes.
don't know if you went back and looked at those families, and then we reported it as or Melissa Bondi's group as a familial glioma gene.
Yes.
And it just came out of the TCGA germline analysis as well. So I think clearly and in none of those other cohorts were they young. None of them were selected for age.
Yeah.
So clearly it has some effect on cancer risk in adults, independent obviously of age. I was wondering in your pedigrees, can you tell, is there any evidence for it getting younger in generations? Because most people don't have that kind of data.
Sharon, thanks for the question. I looked at it like 48 hours ago in a plane. I'm still jet lagged, so I haven't even processed the spelling of POT1, let alone. Sharon Savage raised the interesting possibility that something involving telomeres might be expressed as one age, and maybe your telomeres shorten, and maybe that's one explanation for something that's a little bit odd, because the penetrance appears to be consistent with the dominant effect. We're getting at least the frequency of first-degree relatives affected as if it were something like a P53 mutation, just not at a young age, which is really interesting. And Sharon tells me that it's not seen in osteosarcoma cases, which are a young onset group as a whole, and maybe that's the reason, because that's also a chromosomally unstable disease. Lots to ask.